My new video in which I will be talking about Burkitt lymphoma, and let's start it. Now, Burkitt lymphoma, it originates from centroblasts, and it is the most common non-Hodgkin lymphoma in children and young adults. I've talked about centroblasts in my previous videos. If you check out, yeah, you can say the playlist on lymphomas, yeah, on my YouTube channel. And the pathogenesis, basically there are three kind of presentations, but there is one common mutations which are found in all of these presentations, and that is there is translocation of chromosome 8 and 14. What happens is that a segment of MYC gene on chromosome 8 translocates on alongside the IGH segment on chromosome 14, which results in increased expression of the MYC gene. Generally, MYC gene is expressed in centroblast because all they are rapidly proliferating. But if it undergoes even more rapid proliferation, then it becomes something uncontrolled, and that is what the problem is here, that the MYC region alongside IGH causes increased expression of MYC because IGH has enhancer elements which also you can say enhance the transcription of the MYC gene. And in some cases when we did the molecular typing, we also got that there is increased expression of a transcription factor called TCF3, which was also highly expressed in most of the cases. So let's talk about it. Now what happens that in these three presentations, one is the African one or the endemic one, because you can say the Burkitt lymphoma is endemic in African regions, and one is the sporadic cases which are non-endemic, and the third case has the which is having the poorest prognosis, as the name suggests, is the aggressive lymphoma, in which the patients are also along with the lymphoma, they are also infected with HIV virus. Now what happens is that in the African, you can say the African presentation, there is mutation in the VDJ sequence. Now if I talk about the VDJ sequence, if I take this gene coding for the immunoglobulin, then there's a constant region which is coding for the constant chain. If I draw immunoglobulin here, yes, here, and this is the heavy chain and this is the light chain, then this part, this constant region, is decides the class of the antibody, and this class can be changed or by enzymes. Basically what happens is that when we are producing a perfect antibody, we change the class. We also change one more thing, which is the binding region. You see, the binding region is highly variable, so we also produces change in the VDJ sequence, which is responsible for that. And what it causes is that it we try to, you can say, manipulate the VDJ segments so that we can produce a perfect antibody, because obviously the variable region can be easily changed. That is why it is called variable. So for producing a perfect antibody, we do this. But mutation are mutations, that all right, they are a double-edged sword. They are not, you can say, completely good, they are also bad. So what happens is that sometimes mutations occur, and that leads to, you can say, some unwanted changes or the malignant proliferation we see. So in some cases what happens is that the translocation was 814, but the mutation was involving the VDJ sequence, and in some cases it was mutations were involving the class switch region. Now if I talk about the aggressive lymphoma, you see, in the aggressive lymphoma cases, what happens is that these patients were infected with HIV, and since HIV lowers the immunity of the body, basically what happens is that it depletes the T cell counts. And as you know that lymphoma is also the lymphoma cells, since when they start multiplying, and you can say without any control, they are also acting, or they are also a threat to the immunity. They are threat to our body, so they are also threat to our immunity, and the body tries to kill them. So if a person whose immunity is also lowered and the T cell count is less, obviously the lymphoma will be highly unchecked, and it will grow out of its limits, and it will be aggressive. Now if I talk about the MYC gene, so the MYC gene has a very huge role in Warburg's metabolism. In my previous videos I have talked about that it does increase the proliferation, help in increasing the proliferation of the cells. But how? So in Warburg metabolism, what the MYC gene does is that it increases the synthesis of glycolytic enzymes, and it also increases the shunting of the mitochondrial metabolites. And by increasing the glycolytic enzymes, a lot of glycolytic intermediates are produced, which can be shunted to different pathways. You see, pentose phosphate pathway, you must be remembering that we produce ribose 5 phosphate through that pathway. Now, what is the role of ribose 5 phosphate? Yes, you're right. It is used in synthesizing pyrimidines and purines. Now, if I talk about this citrate, it is going for fatty acid synthesis, and if I talk about oxalic acid, it is going for amino acid synthesis. So basically you can see that everything is going in the part of synthesis. Why synthesis? You must be seeing this kind of scenario in cells which are highly multiplying. So that is why Warburg metabolism is essential, but it does not occur every time. It occurs only in cases when the cell has to proliferate or when it is a blast. But what happens is that due to the increased expression of MYC gene, the scenario is always like this, and this is Warburg metabolism. And also, there is an enzyme here which is called pyruvate kinase in between here, which kind of is, you can say, it converts something to phosphoenol pyruvate and then it goes to pyruvate. So what happens is that the MYC gene also inhibits that. Now, why does it inhibit that? See, if the glycolysis pathway continuously goes into the pyruvate part, then these intermediates won't accumulate that long. You see, in the reaction, if I keep on feeding the reaction and there is no, you can say, stoppage point, then all the intermediates will be consumed. But if I kind of inhibit one reaction or keep it in check, then these intermediates, which are essential, these glucose 6 phosphate or fructose 6 phosphate, which are going for synthesis, these will start accumulating. So that is why this is the role of MYC. Now, if I talk about the clinical features, we see fever and night sweats, and there is extranodal presentation. Now, in the endemic region, in the endemic lymphomas and in the sporadic lymphomas, the clinical presentation is different. In the endemic lymphomas, we see mandibular presentation, most commonly mandible presentation. You can see this kid here, and this is an African kid, and you can see that the mandibular presentation here is very prominent. So we see that here, and there is also involvement of abdominal viscera, in which there is, you can say, the tumor spreads, or you can say the lymphoma cells spread to the kidney, ovaries and adrenal gland. And in the sporadic cases, they spread in the ileocecal region. Now, this, if this is the ileum, you can say intestine has three parts: duodenum, jejunum, ileum. So if this is the ileum, then it connects to the cecum, and this, the cecum part, this here is the ileocecal region. So now intestine goes like this. You must be remembering from human anatomy. So this is the ileocecal region, and that is also involved, and the peritoneum region is also involved. Now, if I talk about the bone marrow spread is very rare. And one more question, but why mandible is involved? This is a kind of a, you can see, unique presentation which we did not see in other lymphomas, which are also having extranodal presentation. So basically this is related to EBV infections. Now, Epstein-Barr virus are those viruses which have a tropism, or you can say selectivity, for oropharyngeal lymphoid tissue. And what they do is that they induce mutations and are responsible for carcinogenesis also. And they also cause uncontrolled proliferation in B cells, which has already, if a cell is already proliferating, it has a higher chance of accumulating mutations or getting mutations introduced into its genome. So that is what EBV causes. Now, if most of the cases, what happens is that if these B cells or the centroblasts present in the oropharyngeal lymphoid tissue, they get infected. So then yes, they are mutated, so they start dividing. Undergo uncontrolled proliferation. And what is the bone which is very, you can say, adjacent to the oropharyngeal lymphoid tissue? Yes, you got it right, that's the mandible. So yes, it goes into the mandible, and also the mandible is very active. If I remember, then maybe it also involved in the production of, you can say, it is a hematopoietic kind of a region. It is a kind of a hematopoietic region. So this also establishes the tropism, that why mandible is more commonly involved. It is due to the EBV part. And if we talk about the biopsy, see, if we take a biopsy from the extranodal side, what we see is that a lot of centroblasts, like I talked about, that they have a appearance in which they are having a multiple nucleoli, and the chromatin is a bit loose, it's not tight, because they are transcriptionally active. And you see the starry sky appearance, the characteristic starry sky appearance, in which the macrophages have engulfed, since the cells are highly, they are having a rapid cell turnover and they are dying, so they are actively consumed by the macrophages, and that too at the cell turnover is not at a normal rate. That is why they are being easily, you can say they are continuously being consumed by macrophages, and these macrophages are consuming the apoptotic cells, give the starry sky appearance. Now if I zoom it in here, you will not able to see it, but what we see is the granular chromatin. Now the chromatin here is very highly expressed, and it is surrounded by a lot of transcription factors, or you can say transcription factor proteins, which are actively expressing it, or expressing the genes on it due to the MYC gene mutation. So due to being surrounded by these transcription factors, the chromatin appears granular. Now if I talk about the bone marrow aspirate, so we see royal blue cytoplasm. We see these cells which are not having that multiple nucleoli. You see a single nucleus, but you're not seeing nucleoli, and they have royal blue cytoplasm and vacuolated cytoplasm. Now these are the Burkitt lymphoma cells only, but why have they suddenly changed? So the reason is that because you see they are not having that stress of dividing in here, so they are having that mutations. But what happens is that due to the microenvironment of the bone marrow, maybe you can say that in neoplasia you must have heard that the microenvironment also plays a very influential role in the growth of the cancer. So the microenvironment provides a very stable environment in which the cell kind of sees that okay, now I will divide, I am malignant, but it's okay, I don't have to divide so continuously, I am getting what I am getting. So uh, also you can see the nutrients it is also getting is very sufficient. Here what was happening is that it was continuously dividing, so these lipids which it was producing, or the proteins it was producing, it was continuously being exhausted. But now what is happening, that since it has got a very chill place, you can say in the bone marrow, it is quiet, it is kind of quiet, so the lipid are being able to accumulate in its cells, and that is what we see. It is dividing, it's not, I'm not saying that it is not dividing, but the microenvironment has kind of simulated it in that way that it does not have to divide that much rapidly. So we see you can say vacuolated cytoplasm, and the blue cytoplasm is because of the increased expression of RNA. Because since see it is still rapidly dividing and it is producing RNA now, because MYC gene is causing it to produce a lot of proteins, and for producing proteins you need the expression of RNA. So yes, the RNA is being produced very much, that is why we see a blue cytoplasm. Now if I talk about the IHC, you see the CD19 and CD20 is positive, BCL6 is positive, but BCL2 is negative, which also explains that why the cell turnover is very high. You see, since it is proliferating very much, it has very higher chances that during dividing it will accumulate mutations and some unhealthy mutations too. And what happens is that the BCL2 protein, it won't stay like that for long, since it is you can say the BCL2 is basically BCL2 what it was an anti-apoptotic protein. So since this is negative, it can't save it from apoptosis. So it will die, it will easily die. And if it accumulates mutation, what happens is that there is not complete suppression of TP53. So at a certain point, these cells die, and that is why you see that the increased mitotic rate also you can say leads to the cell death and the increased cell turnover. So everything is related and everything is logical. So, and yes, just like I told you that this is the presentation which we see here in the endemic Burkitt lymphoma especially. So I end my video today. If you like this video, give a thumbs up, comment down the comment section, and please subscribe. And if you want more, then uh, more is on the way. I keep producing these videos because the views and all the likes keeps motivating me to make even more. I will see you in the next video. Till then, bye bye.